All right, good morning, everyone. My name is Madison Parrinello, and I'm from Regina High School.
My name is Claire Wu, and I'm from Troy High School. And today our presentation is titled, Baby, You're Stressing Me Out. And we know the title is a little cheesy, but today we're going to be talking about babies and stress. More specifically, we'll be talking about how pregnancy-related stresses affect the uterus, especially on a molecular level. All right, so Madison and I are in Dr. Condon's lab, and since our results are closely related, today we're doing a joint presentation. So our lab works on defining the mechanisms that result in preterm birth. And this research is really important because here in the US, we have the 6th highest rate of preterm birth on the planet, with 1 in every 10 babies being born preterm.
At a local level, these rates of preterm birth are even higher. Detroit, in particular, has a preterm birth rate of 18% within the Black population. Similar rates exist in Pakistan, Zimbabwe, and Malawi.
Okay. So during pregnancy, every uterus experiences several stresses, and these stresses can be physiological, chemical, or even hormonal. So examples of these stresses are hypoxia, mechanical stretch, hypertrophy, inflammation, cytokine fluctuation, and hormone fluctuation. So to elaborate on these stresses, hypoxia is low oxygen levels. Mechanical stretch occurs because the uterus has to stretch in order to accommodate the growing fetus. Hypertrophy is increased cell size, and it's the predominant method of uterus growth during pregnancy. It occurs as a result of mechanical stretch. Inflammation is an immune system response, and it's the body's reaction to the fetus implanting. Inflammation is also needed for parturition. Cytokine fluctuation occurs because cytokines are mediators of the inflammatory response, and as we just talked about, inflammation is necessary during gestation. Lastly, we have hormone fluctuation. And this is because hormones like estrogen and progesterone fluctuate throughout the duration of a pregnancy. So in the pregnant uterus, normal pregnancy-associated stresses activate the unfolded protein response, which prevents the onset of premature uterine contractions and therefore preterm birth. So we'll talk more about the unfolded protein response in the upcoming slides. So the endoplasmic reticulum is the site of the unfolded protein response, and the unfolded protein response The endoplasmic reticulum exists within almost every single cell of the body. It has several functions, such as within protein synthesis, protein folding, lipid synthesis, calcium storage, and detoxification. Our lab is most interested in the endoplasmic reticulum's role in protein synthesis and folding. So when protein synthesis or folding is impaired, there's an accumulation of misfolded or unfolded proteins, and this leads to endoplasmic reticulum stress. Since the cell's goal is always to maintain endoplasmic reticulum homeostasis, this stress must be resolved. And the cell does this by triggering the unfolded protein response. So the unfolded protein response has several constituents, the first of which is expanding the endoplasmic reticulum by increasing lipid synthesis. And this works because the ER is composed of lipids. So the second option for the cell is to attenuate protein synthesis, which reduces protein load. Thirdly, the cell can remove misfolded proteins through ER-associated degradation. And the cell needs to do this because misfolded proteins are non-functional. And lastly, the cell can produce chaperones and protein processing enzymes. Chaperones help protein folding, and both of these are essentially expanding the machinery to fold proteins. So there are 3 main pathways that ER stress triggers and that constitute the unfolded protein response. So each of these pathways starts with the transmembrane protein that's embedded within the endoplasmic reticulum. And these 3 pathways are IRE1, ATF6, and PERK. So for the IRE1 pathway, XBP1 becomes spliced. And this spliced version of XBP1 goes on to act as a transcription factor and is able to upregulate the production of chaperones, increase lipid synthesis, and also trigger ER-associated degradation. So once again, ER-associated degradation removes misfolded proteins from the cells. The second pathway is the ATF6 pathway, and ATF6 becomes cleaved. This cleaved version is able to upregulate genes coding for ER chaperones and ER-associated degradation components. Lastly, we have the PERK pathway, and it results in the phosphorylation of EIF2 alpha. The phosphorylated version of EIF2 alpha is able to cause general inhibition of protein translation. CHOP upregulation also results from the PERK pathway, and CHOP induces apoptosis or cell death by eventually activating caspases. So in most cells, CHOP causes apoptosis. However, in pregnant uterine cells, non-apoptotic caspase 3 becomes activated, which helps maintain uterine quiescence. And uterine quiescence just means that the uterus is not contracting before it's meant to. All right, so the three pathways that we just discussed are typically kept inactive by the three transmembrane proteins binding to BIP, and BIP is a chaperone protein. So when the cell encounters ER stress, BIP is able to dissociate from the three transmembrane proteins and instead bind to the misfolded proteins. So this has two functions. First of all, it activates the three pathways to help reestablish ER homeostasis by triggering the unfolded protein response. And secondly, it prevents the misfolded proteins from aggregating, which causes harm to the cell and to the body. Okay. So these types of stresses that we just previously discussed can either be local or systemic. So we wanted to identify if uterine quiescence and the unfolded protein response are regulated locally or systemically. So if they're regulated systemically, things like cytokine fluctuation and hormone fluctuation would be controlling these processes. On the other hand, if they were regulated locally, things like hypertrophy or mechanical stretch would be controlling these processes.
All right, so these types of stresses that were just previously discussed can either be local or systemic. You can see in the picture to the left, the effect on the fetuses of the effect of the fetus on the mom's pregnant uterus would be considered a local type of stress. Whereas on the picture to the right, pregnancy-associated circulating factors affecting the mom's uterus would be considered a systemic stress. Therefore, it would be something involving the blood of the pregnant mother. And we have determined which uterine proteins are affected in a systemic or local manner during pregnancy in the next few slides. So the next graph is changes in estrogen or progesterone. An example of systemic factors would be these two hormones. These are different types of chief hormones fluctuating throughout a rat uterus during pregnancy in the graph. Now, looking at the dotted line, you can see that progesterone decreases towards term, while estrogen increases towards term. These hormones are vital considering that estrogen enables the uterus and placenta to move, causing vascularization, which means it helps with the formation of blood vessels. While progesterone plays a role in preventing the uterus from contracting early. So therefore, these would be systemic factors. So here's the methods portion of our presentation. All right, so our lab, we use the unilaterally pregnant rat model to examine the effects of these local and systemic factors. This model was generated by taking a non-pregnant rat, which is a rat with a biocarnate uterus, meaning it has two uterine horns. We tied off one horn just below the ovary so that when mated, the eggs from the tied off uterine horn will never get fertilized. And you can see in the picture to the left, the little string. So once we tied those off, they would never get fertilized, while the other unligated horn does get fertilized. These tissues were then isolated from the empty unfertilized horn and the gravid fertilized horn and cleared of all fetal material. Cytoplasmic and nuclear proteins were extracted and probed for the proteins of interest by Western blot analysis, and these samples were collected from gestation day 6 to gestation day 23, which is term for pregnant rats. We then examined these tissues for markers of the unfolded protein response and inflammation and prostaglandin synthesis, all crucial components that regulate the onset of uterine contractions at term. So in this model, both the empty and gravid uterine horns will be subjected to the circulating systemic changes associated with pregnancy, but only the gravid uterine horn will experience the local fetal-driven consequences. And to the right, you can see an example of a human uterus. So here's the Western blot protocol that we followed for our experiments. So first, we would run a gel by loading a sample into each well, and once the gel is running, the proteins separate based on size. And you can see to the left is the gel running, on the bottom is all the proteins, and then to the left is called a ladder. So next we perform a transferring process in which the proteins in the gel are transferred onto a membrane, and the only visible colors on the membrane is the ladder part, in which serves as a marker for the protein so we know where to cut. So as shown to the right, you can see the membrane. So after the transferring is done, we block the membrane using a milk buffer to reduce antibody and background proteins on the film. Once the membrane is blocked, we apply primary antibody, and that will only bind to our protein of interest. Next, we apply secondary antibody, and this binds to our primary antibody. And the final step is to apply chemiluminescence, in which will bind to the secondary. So lastly, we pick up the signal from the chemiluminescence by placing a film onto the membrane, developing it, and you can see our film results in the next.
So this is the Kumasi blue stain gel. So it just shows the proteins that are remaining on the gel. Oops, okay. So first of all, the goal of a Western blot is to examine differences in expression of a certain protein across different tissue samples. And these tissue samples may be treated differently or be from different time points. Essentially, they show some sort of manipulation in the independent variable that you want to test in the experiment. So loading controls are proteins that have high consistent expression across all the samples being studied. And loading controls have a few roles. First of all, they quantify the amount of protein loaded in each well. And secondly, they check if proteins were evenly transferred from the gel to the membrane. So essentially, they normalize unintended variation, such as due to differences in loading between the different samples. So on the bottom of this slide is a Western blot for PDI. And the Western blot was performed on samples prepared from uterine tissue from the unilaterally pregnant rat model. Tissue was collected from day 6 to 23 of gestation, both from the empty, unfertilized, and gravid fertilized uterine horns. And PDI proved to be a good loading control, as evidenced by its relatively consistent expression from day 6 to day 23 of gestation. There's also similar levels between the empty and the gravid uterine horns. All right, so these proteins on this slide are not suitable to be used as loading controls. So we have tubulin, calponin, SRC3, and GAPDH. And they're not suitable to be used because you can see that protein expression changes from day 6 of gestation to postpartum. We also see differences between the empty and the gravid uterine horns. So interestingly, in other species, these proteins have proved to be good loading controls. So we can see that there's a lot of differences between a rat pregnancy and a human pregnancy, for example.
All right, so next is the results part of our presentation. So this diagram portrays how prostaglandins, a type of lipid compound, are made. So when prostaglandins are produced, the uterine compartment, they promote uterine contractions. You can see that caspase-3 activation, which is written in red here, leads to phospholipase A2 cleavage, generating arachidonic acid. The conversion of arachidonic acid to prostaglandin depends in part of NF-kappa B activation, as you can see in the diagram to the right. So we know that the unfolded protein response prevents prostaglandin production earlier in pregnancy by maintaining caspase-3 in a non-apoptotic status. And we also know that the unfolded protein response can limit inflammation, again, acting to limit prostaglandin production. So therefore, the woman does not go into labor. So increase of prostaglandin leads to increase of contractions. Therefore, we looked at the components of the unfolded protein response, BIP and CHOP, to see how these proteins were regulated. Okay, so on the graph, you can see BIP and CHOP here. So from early gestation, BIP upregulation only occurs in the gravid uterine horn, suggesting its upregulation is dependent upon the presence of the fetus, whereas in the empty uterine horn, it remains unchanged. So in contrast, CHOP levels in early gestation are very high in both the empty and gravid uterine horns, suggesting that CHOP activation is regulated in a systemic manner. However, from gestation day 17, we see a sharp decline in CHOP in the gravid uterine horn, with its decline to undetectable changes in both uterine horns as term approaches. This data demonstrates that BIP is likely regulated by the presence of the fetus, whereas CHOP is likely regulated by circulating factors. So also in the graph, you can see the light blue bar is our empty, while the dark blue bar is our gravid. So BIP is a cytoplasmic protein. So PDI was used as our loading control, and CHOP is a nuclear protein. So full-length PARP was utilized as our loading control. And we utilize these as our loading controls because their expression remains pretty stable across gestation. So the next protein is caspase-3, and you can see at gestation day 16, they are regulated equally in both the gravid and uterine horns. However, throughout gestation to term, the gravid uterine horn is highly active for non-apoptotic caspase-3 activation. So as discussed earlier, active caspase-3 activation is normally associated with apoptotic cell death. However, in this model, cleaved PARP is isolated to the empty uterine horn and never appears in highly caspase-3 positive gravid uterine horns, as measured by PARP cleavage on the second plot. This indicates that caspase-3 in the gravid uterine horns is non-apoptotic.
Yeah.
So in the section, along with the increase in caspase-3 levels to term, we also observe IPLA2. which is a hallmark for increased prostaglandin production, as I mentioned earlier, which occurs solely in the gravid uterine horns. So overall, this demonstrates that caspase-3 activation and downstream prostaglandin production are both dependent on the presence of the fetus and not regulated by circulating factors. Therefore, they are considered a local factor. And you can see below, we have PDI as our loading control, and then above is the protein IPLA2.
Okay, so the second section of our results has to do with the role of inflammation throughout gestation. So to begin the pathway seen on the slide, we have toll-like receptor 4. Toll-like receptor 4 goes on to activate phospho-TBK1, and phospho-TBK1 activates transcription factor NF-kappa B. NF-kappa B causes inflammation, so you can see how this is an inflammatory pathway. And NF-kappa B also increases prostaglandin production. Since prostaglandins are what trigger uterine contractions, NF-kappa B helps regulate the duration of pregnancy. So the unfolded protein response that we discussed earlier actually inhibits NF-kappa B's role in prostaglandin production, which holds off labor. So essentially, during the earlier stages of pregnancy and throughout the most of pregnancy, the unfolded protein response triggered by gestational stresses prevents premature labor. So we mainly focused on the first three proteins seen in this pathway: toll-like receptor 4, phospho-TBK1, and NF-kappa B. All right, so first we have NF-kappa B, and as we saw in the previous slide, it mediates gestation by causing inflammation and also activating prostaglandin synthesis. NF-kappa B targets inflammation directly by increasing production of cytokines, which help mediate the inflammatory response. So with the Western blot of NF-kappa B, which you can see on top, we normalize the data with loading control PARP. Specifically, we used full-length PARP, which you can see the arrow pointing to. We then put the normalized data into a graph with the empty uterine horn being indicated with light blue and the gravid uterine horn being indicated by dark blue. So we can see how NF-kappa B is upregulated in the gravid uterine horn every day after day 6 of gestation. So this indicates that the presence of the fetus somehow causes a local increase in expression of NF-kappa B. And if we take a look at day 23 specifically, we can see how NF-kappa B is especially elevated in the gravid uterine horn. And this is because inflammation, which NF-kappa B leads to, is essential for parturition or giving birth. So next we have TLR4 and phospho-TBK1, which are upstream of NF-kappa B and therefore also help mediate inflammation and prostaglandin synthesis. So we normalize the data from the Western blots of TLR4 and phospho-TBK1 with loading control PDI. We then put this normalized data into graphs with, once again, the empty uterine horns being indicated by light blue and the gravid uterine horns being indicated by dark blue. So toll-like receptor 4 in other species has been described to be upregulated at the end of pregnancy to allow for increased inflammatory activation. And we observed something really similar here in the pregnant rat. So in the pregnant rat, TLR4 levels increase towards term in a gravid horn specific manner, showing that the presence of the fetus causes a local increase in expression of TLR4. TLR4 and phospho-TBK1 show similar patterns of variation across gestation, due to phospho-TBK1 being directly downstream of toll-like receptor 4. And you can see the similar pattern in both the Western blots and also the graphs. So they all have relatively high expression in early gestation, lower expression in the middle of gestation, and are again upregulated towards the end of gestation, especially in the gravid uterine horns. So in early gestation, both TLR4 and phospho-TBK1 show high levels of expression because of the inflammation that implantation of the fetus causes. If we take a look at phospho-TBK1 specifically, we can see a really elevated protein expression in the gravid horn of day 23. So this indicates that the bulk of inflammation occurs right as the rat gives birth. All right, so moving on to our conclusion slide. So our analysis demonstrates that many of the proteins in the prostaglandin pathway, such as TLR4, IPLA2, and phospho-TBK1, are all regulated locally by the presence of the fetus. So the presence of the fetus through things like mechanical stretch or hypertrophy causes local responses in late gestation, especially in days 21 and 23 of embryonic development. So if you look to the bottom right of the slide, you can see TLR4, IPLA2, phospho-TBK1, and you can also see day 21 and 23 of gestation being indicated. So the unfolded protein response serves to inhibit the production of prostaglandins, and the proteins involved in the unfolded protein response appear to be regulated in many different ways. BIP is one of these proteins, and it seems to be locally regulated consistently throughout gestation, with the gravid uterine horn always showing higher levels of expression than the empty uterine horn.
So on the other hand, CHOP appears to be systemically regulated for the majority of gestation. CHOP is elevated in both uterine horns early in gestation and declines to undetectable levels in both uterine horns at term. Circulating hormones such as estrogen and progesterone, which change as term approaches, or circulating inflammatory cytokines could be the mechanisms behind CHOP regulation. So this analysis increases our understanding of local versus systemic changes that occur during pregnancy and reveal the dynamic interplay between systemic and local changes that will likely work together to promote an appropriate gestational length and the prevention of premature birth.
All right, so next we have some future research possibilities. So we know that CHOP is regulated in a systemic manner, potentially by hormones like estrogen and progesterone. So since progesterone typically decreases towards the end of gestation, a possible research idea is to keep progesterone levels high throughout pregnancy and examine the effects that has on CHOP expression. Similarly, we could keep estrogen levels constant throughout gestation to examine the effects estrogen levels have on CHOP expression. So a second, more general research idea is we can further explore the factors that affect NF-kappa B expression across gestation. And this is because both TLR4 and phospho-TBK1 are directly upstream of NF-kappa B, yet show a slightly different pattern of variation across gestation. So research could be done in this aspect.
All right, we'd like to thank Dr. Condon, Dr. Surya, Francis, and Wayne State University and the Discovery to Cure program for giving us the opportunity to be able to present and learn in our research.